So we're getting ready to go on a very long journey. And that journey is in the world of protein structure. And before we take off on this journey, we're going to have to understand what we would be seeing once we look out of those airplane windows. And there's going to be a couple of new definitions that we're going to talk about, and one of those is called the type of protein structure that we're regarding. And there's a couple of different ones. So we're going to first start there, and we're going to expand upon our vocabulary a little bit, and we're going to see primary and secondary and tertiary and quaternary structure. And these all have abbreviations, just like we have always used in organic chemistry: primary, secondary, tertiary, and quaternary protein structure. So we're going to take a look at each one of these, and we're going to discuss a little bit about what makes them different. What is really primary protein structure? What's really the secondary? What's tertiary? And what's quaternary? And we're just going to kind of build up on top of those definitions from that point on. And we're going to start talking about how to analyze proteins now, not just simply amino acids. So we're kind of building up from a foundation like we always have and like we always do. So let's first start talking about primary protein structure. So if I look at primary protein structure, this is basically the sequence of amino acids. That's all that it is. So if I want to know information about the primary protein structure, what I'm after is just really simply the amino acids and the order at which they hook up. That is all that I want to know. So what amino acids are there, how are they hooked up, which one's first, which one's second, which one's third, and which one's fourth. That's all that primary sequence is. So amino acid number 1 hooked up with amino acid 2, hooked up with amino acid 3, and then I'm going to put ... because that can continue on. So that is primary sequence. If the question asks for primary sequence, that is what they're after. They want to know what amino acids, which ones, how, when, where, why, and how. The secondary protein structure is a little bit different. The secondary structure begins to start taking a look at these amino acids, and it begins to answer the question of how they fold. And fold basically means begin to take shape. Okay, so that's very important. Protein shape is very important. So when I take a look at secondary structures of proteins, what I'm looking for is what kind of shapes are they bringing to the table. And there's two different types of secondary structure here. There is one that we call the alpha helix, and then there's one that we call the beta pleated sheet. So the alpha helix is basically like a slinky. So this is maybe some of the shape that secondary proteins or the secondary structure of proteins could describe. And a beta pleated sheet is basically just a sheet, kind of like a flat layer, and they're pleated. So it's kind of like an accordion file in a way. If you want to think of it that way, imagine you taking a sheet of paper and imagine you folding it into an accordion. So that is what a beta pleated sheet is. Typically flat, there's some creases in it, you're making these pleats, you're making these folds, and that is what happens with beta pleated sheet. So these are the two of the secondary structures that we're really going to be focused on during the lecture. The next type of protein structure is going to be called tertiary. And tertiary is more folding as well. So we can take a look at tertiary structure, but really when we take a look at the folding, we're looking at the 3D shape finally. So alpha helix and beta pleated sheets, those are really only kind of on the surface. So what are the basic type of shapes that we are seeing in the protein? And then the tertiary, there's a 3 in front because you need to think of 3D. once this protein does all of the alpha helix and beta sheets that it wants to, how is it actually ordered in a three-dimensional space? So what does it look like? Is it globular? And if so, what kind of specific shape does that globular protein look like? And then finally, we've got a quaternary structure. And this is more folding, but quaternary structure is even more specific because it's basically focusing on more than one 3D shape. So what we have here in quaternary is a 3D shape from multiple pieces. And we want to know what those multiple pieces are doing with each other. So there is the difference between primary, secondary, tertiary, and quaternary. And we're going to take a look at each one of these in a little bit more detail and talk about really the logistics behind each one. And we're going to start with primary first, and then we'll go to secondary, tertiary, and then quaternary is how we'll end the lecture and the video series up. So we're going to first start looking at primary structure. And primary structure we've kind of already talked about up to this point. We've taken a look at amino acids, we've taken a look at how they order themselves up, how we can protect the N-terminus, how we can activate the C-terminus, and really the importance of how the amino acids are ordered. Very, very important. Everything is determined by the order of the amino acids. So we need to take a look and maybe discuss a little bit about primary sequence. And that is what we will do in the next video. So we're going to take and we're going to look at a different perspective here. What we're trying to end up accomplishing with this whole mess and this whole description is we've got a blobby, maybe protein here. And we want to know what amino acids are making that protein up. And there's a couple of different ways that we can do that as far as reagents and enzymes go. So the primary protein sequence, or the amino acid sequence, will look at this protein, it will rip it apart, and it will kind of go through step by step of how these amino acids are hooked up and in what order they're hooked up in. And that's what we'll do in the next video, so stay tuned. There's the definition, at least, of primary, secondary, and tertiary.